Okay, so over to you, Wim.
So thank you very much for the introduction. And I would like to share my screen, but it seems the host disabled the screen sharing. So I do not know if you are able to allow me to share
that out.
screen.
There we go, more.
I try again. No, it's
Make
still...
a host. Try again, Wim.
Yeah, I will try again now. Okay, so let's see. Share.
There we go. We can see it.
Do you already see?
Perfect.
Okay, just activate my laser pointer. Okay, so good morning, everyone. Welcome to the next lecture. So as John already told you, so my talk will focus a little bit on environmental stress by pesticides. And so we will rather explain how early life pesticide exposure, like prenatal pesticide exposure, can have long-lasting effects in the offspring child or later in life also, if the pesticide exposure is continuing. So this can also contribute in adverse epigenetic metabolic programming. And so we will see an example of how this can be linked to cardiometabolic risks and NAFLD liver pathology. So I will first briefly give some background on what are causes of obesity and NAFLD or definitions. So then I will also briefly explain some biomarker studies. So biomarker approaches in cohort studies and what's the difference between genome-wide association studies and epigenome-wide association studies. So how you can link this to disease risk. And then I will just briefly give an overview on how pesticides can trigger adverse effects in children. And then we will do kind of a validation study in an obese cohort or in a NAFLD cohort, where we also looked at links between, where we try to validate some of the links that we have identified in the first study. So as most of you may know, so we observe like an increasing prevalence of obesity worldwide. And in the last 30 years, the number of obesity cases have nearly tripled, as you can see, between 1975 and 2016. So and people are wondering, so what are causes of this increase in obesity? Obesity is a complex disease. So like 15 to 20 years ago, people were convinced that we would find all explanation in obesity disease by genetic mutations. And I will come to that back in a few slides. So definitely we cannot explain all obesity variation or all variation in metabolic phenotypes just by looking at the genetics. So there's an also complex interplay with lifestyle. So are you stressed? Are you not stressed? Do you have sedentary lifestyle? Do you walk a lot, or do you do a lot of sport? So you suffer a lot of mental stress in your environment. Also, what is the kind of food you consume? So it all has to do also with like the energy intake or the energy expenditure. So it seems like mitochondrial metabolism also plays an important role in some of the obesity phenotypes or in the epigenetic programming. So if there's an imbalance in energy intake as compared to energy expenditure, so then you are at higher, you have higher risk to develop like obese phenotypes. So in general, if we talk about obesity, it means that there's a kind of imbalance in energy consumption and energy expenditure. Another thing is that if you look to what is the contribution of the genetic component, for example, if you have tall parents, then the chance that you have tall children are very big. So 80 to 90% of the children of tall parents will also be tall. But if you look to the metabolic phenotypes, there's a much broader variation in phenotype or in weight or in fat deposition there's only 35% of genetic component from the parents, and then there's about 65% of environment, which contributes in variation of the obesity phenotype. And yeah, since I will talk about pesticides, so it's not only the food exposure and energy intake by food, but also endocrine disruptors and pesticides. So they can also manipulate the hormone pathways, and they can also trigger some adverse effects in cells which contribute to the obesity phenotype. So if we talk about extreme obesity, then we calculate the BMI, and if you have a BMI value of above 25, you are considered like an obese phenotype. So I'm not a physiologist or a medical doctor, so I forgot to look up how it's calculated. But yeah, most of you are familiar with BMI values. So, and you see with increasing BMI value, you are more close to the obese phenotype. So why are we interested in trying to reduce obesity? This is because it can lead to many comorbidities. And later on in this talk, we will look to comorbidities linked to cardiovascular disease, but also liver fat disease, like the non-alcoholic fatty liver disease. So beside this, there's other comorbidities like cancers. So because obesity also can create a pro-inflammatory environment, and this can accelerate aging and then also accelerate aging disease like cancer, neurodegenerative disease, and some of the metabolic diseases. So, and later on in my talk, we were interested in to find links of pesticide exposure to obesity phenotypes and NAFLD phenotypes. And as you will see, that the adverse epigenetic effects do not only depend on the environmental exposure, but the adverse epigenetic effects will depend also on interplay with your genetic configuration or with some genetic polymorphisms. Okay, so I already told you that the genetic configuration, your genome, does not explain all the heritability of obesity phenotypes, and that the environment also plays a contribution in the phenotypes. And if you look to this scheme, so we have to explain a little bit what kind of associations you can look for to explain obese phenotypes. So in the last 20 years, there's many research studies which have defined genome loci or genes, which like variation in genetics, genetic changes in genes, which can increase the risk for obesity. So, and we call them the genome-wide association studies. But because now we are more aware that also lifestyle factors can play a role in obesity, so also people have looked to by epic arrays, by epigenome-wide association studies. So you look to association of methylation patterns, which are more prevalent in obesity phenotypes. So these are two concepts which you may already be familiar with, but there are still some other biomarker studies ongoing in obesity studies. And this is, for example, they look for EQTLs, and I will also explain in a minute. So these are genetic variations or polymorphisms which change expression of genes, and this variation in transcriptome data then can also be linked to obesity phenotypes. And then you have another possible biomarker, and we call them the methylQTLs. So it seems that some genetic changes or polymorphism can impact the DNA methylation pattern. And so then it looks like the genetics and the epigenetics is tightly linked and interdependent. So, and here there's like also an environmental component which can link the genetic polymorphism to the epigenetic consequence. And we will discuss this also during this talk. So, and then in this way, epigenome-wide association studies may also have like a genetic component. And this is also something that I will explain in a minute. Then, how people are identifying all these biomarkers. So you need to start with cohort studies, and then the kind of cohort study that you're working with also has advantages and disadvantages. So the most popular one is that you just take a group with non-obese people and obese people, and then you isolate the DNA and you do like a genome-wide association study. You look to genetic variants, or you do epic array, and then you do methylation variation, and you try to link this to the obesity phenotype. So here you will have a lot of noise because you have a genetic, very heterogeneous population. And also you will have on top of that a lot of epigenetic variation. So here it's very difficult to really have robust biomarkers in a population linked. So you need, in general, you need like quite large cohorts to have enough statistical power to identify the biomarkers of interest, either the genetic biomarkers in genome-wide association studies or the epigenome biomarkers in epigenome-wide association studies. So typically you will see nowadays, you see this kind of studies with thousands of participants. But okay, sometimes this is not really accessible. If you're a standard academic lab, you may not have enough budget to deal with all these samples and to run all the sample analysis. So that's why people or some labs, they focus more on like well-characterized families where they have already a pattern of inheritance, for example, of obesity. But in general, yeah, these are quite specialized studies also because you have to follow up the parents and the children. And yeah, sometimes it also needs quite longitudinal studies. So these studies or cohorts are not so, there's not so many available, but at least here you can study like genetic inheritance more easily because you have the parents, sometimes the grandparents and the children or the grandchildren. Then you also have cohort studies of twin studies if you have monozygotic twins. So these are the most interesting ones because here you start with twins which are obese or non-obese, and if they are genetically identical, then you can really evaluate very clearly what is the impact of the lifestyle on the epigenetic differences. Also here, it's difficult to have access to these populations. So, and then you also have longitudinal studies. So they are very interesting to look for causal effects, but also it's the challenge here is that you have to collect money to maintain a longitudinal study. So also in Flanders, we have like different studies, Flex 1, Flex 2, Flex 3. So because of the, from the government, they collect prospectively samples from populations in Flanders, and then they use it for follow-up studies to look for environmental impact on particular health effects. But also here it's quite budget. So most of these studies require quite some budget to maintain the studies, either to quite expensive in function of time or quite expensive in the number of samples you have to analyze. And if you look to the history of association studies, either genome-wide association studies and later on epigenetic association studies. So you see here that in the last 20 years, now we have approximately like 900 gene loci which have been linked as risk factors for obesity. But even with this 900 loci, they can only explain a limited percentage of the BMI heritability. Here also you see variations. So lists of genes, they explain here 6%, 17%, 14%. So it seems that you only capture a fraction of the heritability of the obesity phenotype. And then here from later on in 2000, from 2010, people are also entering in the epigenetic field and trying also to link methylation SNPs in the whole obesity disease model. So for genome-wide association studies, you may be familiar. So after the first HEAT genome was published, like you see, there were some hits already which could link these genes to obesity. To increase the power, they went from 1 genome to 1,000 genomes, then to 100,000 genomes, and the more participants include in the cohort study, the more solid, robust some of the markers become, but still you lack some of the heritable, you lack some of the information which explains the obesity phenotype. So then I will briefly explain a little bit the other markers that we will still need in some of the coming slides. So the EQTL is like genetic variation or genetic SNPs which change the expression of a gene. And for example, here, if we think of a polymorphism in a promoter, so this promoter polymorphism could change the binding of a transcription factor and in this way affect the gene expression. And you see an example here for one polymorphism. So depending if you have an A or a G on this position, the gene expression will change. So, and we call this a cis-EQTL because it's in the sequence of the same gene. If we talk about a trans-EQTL, it's a little bit more complicated. So then it could be, for example, a SNP in the coding region of a transcription factor. And this SNP may then change the affinity, the binding affinity to a promoter region of another gene. And then, so then it's like an indirect effect. So this SNP has an indirect effect on gene expression of another gene. So that's why we call it trans, because it binds to another gene. Then also you have a similar trend. Depending on the polymorphism, you see a change in expression of an other gene. Then we come to the epigenome-wide association studies in response to various lifestyle factors. So it can be psychosocial lifestyle factors, or it can be endocrine disruptors, the climate that you live in, or if you live in a city, or if you live in an urban region. So all these factors may also contribute in obesity risk, and this we will see in a minute. And this epigenome-wide association study, most of the epigenome-wide association studies are focusing on DNA methylation because, yeah, in the dogma of the epigenetic, it's like the DNA methylation of particular regions in the genome. They regulate the genes. They can switch genes on or off. And the classical way you analyze it is by epic arrays, but before you hybridize your DNA to the epic array, you have to do the bisulfite conversion, whereas the methylated CpGs are not modified after the bisulfite treatment. The non-methylated CpGs are converted to uracil or after PCR to a T. And then you can, after the bisulfite treatment, you can hybridize it to the epic arrays. So you may have talked already about this in the previous talk, so I will not explain this in much detail. And because we have now hundreds or thousands of epic array signatures or data sets, and we also have clinical data of all the samples, people start to look for patterns which link to obese phenotype. And here you can see these are all CpG probes on the epic array, which are linked to obese phenotype. Like this is like metabolic syndrome, waist circumference, glucose levels, triglyceride levels, the high density cholesterol particles, and then the blood pressure. So for many of the metabolic health outcomes, you see that some methylation patterns are markers for this metabolic risk or disease risk. And you see that some parameters cluster together, and like this one is like the opposite than the other obesity hallmark. So this is like a good parameter. So this is the good cholesterol. So if the good cholesterol is lower, you may be at advanced risk for metabolic syndromes. So, and then I come here to the methyl QTL. What do we understand with methyl QTLs? So these are the polymorphisms in one region which can change methylation in another region. And here, for example, this is a gene melanocortin 4 receptor for which there are many polymorphisms available. And here they show one polymorphism. So, and it looks like the polymorphism at this site determines, has a significant impact on the methylation of this CPG probe, for example, in the neighborhood. And nowadays, because we have a lot of epic array data and also genome sequencing data, like from SNP arrays, they try to correlate SNP variation, so genetic SNP variation, with methylation density variation. So, and these are examples of such studies which have done this kind of analysis, and you can find these correlations. So from all the epic array CPG probes linked to all available SNP data from Illumina SNP arrays. So, and this creates a new kind of biomarker linkage of genetic components which are linked also to DNA methylation changes in the genome. And yeah, my talk, so it's a little bit a long introduction, but this I will start from here to explain how SNPs in one gene, the PON1 gene, can have an impact on epigenetic changes in response to pesticides. And you can have SNP-specific epigenetic variation, and only for one SNP we will develop some cardiometabolic disease. So why is it important? Because SNPs are also present in different ethnic populations. So like many of the biomarker studies are a little bit biased for like Caucasian cohorts. So if you find some biomarkers, it may not always be valid also in another population, like African population or American population or Latin American population. So nowadays you can also do ancestry studies based on methylation SNPs. And this methylation SNPs, the SNPs and the methylation consequence may also determine how we metabolize some compounds. So depending on the presence of some genetic SNPs, you may have different potency in drug metabolization or in pesticide detoxification. And this is what I will want to show you in the coming minutes, that the PON1 polymorphism determines how well you can metabolize or detoxify some pesticides, and the way you detoxify the pesticide early in life or during prenatal life will impact some cardiometabolic pathways in the children or in the offspring. Before I switch to this part, I want to point still to some important differences between the genome-wide association studies and the epigenome-wide association studies. So if we talk about genome-wide association studies, typically a SNP, it's like in a binary code. Either it can have only 3 states. Either you have a wild type without a SNP, or you have a SNP, like a SNP variant. You're an heterozygous, or you have a homozygous SNP. So this has only 3 outcomes. However, methylation patterns, which are linked to SNPs, so this methylation change has like a spectrum. It has a beta value between 0 and 1. So it's like a continuum from 0 to 100% methylation. So this gives more a probability effect, and it has not really a binary outcome. Then also the methylation changes that you identify, they are stochastic and they may accumulate. So depending on the dose and the duration of the exposure, the impact of the methylation change may still increase, which is not the case for the SNP. You have it or you don't have it. But so this is also something to take into consideration, whether the disease risk will increase or not, or is a problem for the disease later in life. And then also the tissue specificity. SNPs you can identify in whatever tissue you collect the DNA from, but the epigenetic changes or the methylation changes may be more apparent in tissues which have been exposed to like the pesticides. So the methylQTL, the SNP impact on a methylation somewhere in your epigenome it may be different in blood samples or liver samples or other tissue samples that you collect. So this can also be challenging. If you need to have some prediction of the disease risk. Okay, and finally, after already I think half an hour I will talk a little bit about the pesticide exposure and about the study that we have conducted. So this was in collaboration with the lab in Denmark, and I think the lab of Helle Andersen in Odense in Denmark. And there they followed a population of women who was working in glass houses. And so in Denmark, there's less sunshine than in Italy. So many of the vegetables are cultured in glass houses. And some of the young female or the women who are working in the glass houses, they were using pesticides at a time when they were not aware that they were pregnant. And then a few weeks later on, or a few months later, when they went to the doctor and it was confirmed that they had a pregnancy, then, for example, they had already a few weeks to a few months exposure to pesticides in the glass houses. So then, of course, they stop with working in the glass house once they know that they are pregnant. But they followed up the females who were exposed to the pesticides, and they had also some control group of non-exposed pregnant female. And then they looked to the children of this population. And then in some subpopulation of the children, they found that there was a problem, a metabolic problem. So that they were more susceptible to metabolic disease, more susceptible to obesity, and also more susceptible to cardiovascular problems. But also there were also children from women who had not really a health problem. And because at that time the lab was also working on polymorphisms, so they are interested in paroxynase 1 polymorphism regulation. So they found out that all the children with this health problem, they had a particular polymorphism. So here, whereas the other polymorphism was much less a problem for the health of the offspring. So what is PON1 doing? So it's like an enzyme which is detoxifying a lot. It's a promiscuous enzyme which is hydrolyzing a lot of substrates. It has lactonase activity, esterase activity. So it's detoxifying drugs like statins. But beside this, it's also detoxifying a lot of other chemicals, including pesticides. So depending on the expression level of the PON1, you may be able to detoxify pesticides and have changed the concentration of the systemic concentration of some of the pesticides. Not only the regulation of the expression, also the activity can be regulated by multiple chemicals or by nutrition and food. So this enzyme is also expressed in the liver and then is secreted as part of the ADL particles. And it also is a kind of protectant for oxidation of LDL particles. It also has some activity, plays a role in inflammation. And of course, the PON1 is also involved in detoxifying systemic compounds. So in here you can see, for example, this is a pesticide, chlorpyrifos, which I'm not sure if it's still allowed to use this one, but it can detoxify this pesticide via the esterase activity. And let's see, so, and the PON1 activity is regulated by both activity and by regulation. And there's many polymorphisms which have been identified in the promoter region, which can affect gene expression. And there are also polymorphisms identified in the coding region, which can change the activity. So in our study, we have looked to different kind of polymorphisms and what is the impact on the outcome. So here you see the impact of the polymorphisms in the coding region, which will change the conformation of PON1 and has an activity. That will change the enzymatic activity. And then here, there's also like a polymorphism in the promoter. And yeah, as you will see in the coming minutes, it can also affect the methylation status of the promoter and the gene expression. And then finally, besides the polymorphism that we have identified, the PON1 enzyme is also involved in the methyl donor cycle. So it's linked. So depending on the PON1 activity, the concentrations of methyl donors will also change. And so it may also impact DNA methylation regulation. So you see it's a complex, there's many levels of regulation. And as you can expect, depending on the PON1 expression level in response to pesticide concentration, you may have complex changes in the downstream epigenome. So here I mention a few examples on what are possible adverse effects of the pesticides. So you see that exposure to pesticides in combination with some PON polymorphisms has been linked to some cognitive problems, also to obesity problems and cardiovascular problems. So, and what we did is, so we collected like DNA samples from the children to see if we can detect a difference in epigenetic patterns in exposed and unexposed, like in the offspring children from mothers which were exposed or unexposed to pesticides, and then in a different background, so with different SNP polymorphisms. And okay, to make a long story very short, so this is like a heat map of the epic arrays. And what we could observe is that we have like a synergy in epigenetic changes in combination with pesticide exposure only in a subset of particular genetic SNPs. So it seems that the impact of the pesticide is highly dependent on the particular SNP of PON1, which is involved in the detoxification of the pesticides. And what we could observe is that we observe a lot of changes in neuroendocrine pathways like dopamine pathway, corticotropin pathway, neuroregulin pathway. And if we look further, this neuroendocrine pathways, they regulate like the eating, the appetite and the eating behavior. So they regulate the food intake and the energy balance. So it looks, this was an important component. And also we find that these neuroendocrine pathways are critically involved also in many of the metabolic disease that we observe, like obesity, diabetes, here cardiovascular disease. And also here we find links with the mitochondria, the mitochondrial function. So, and then we also look for associations of particular genes with the PON1 activity, and we find that depending on the SNP, we have differences in PON1 activity, and we also find many links to the genes. So SNP-dependent changes in PON1 activity and DNA methylation of target genes, neuroendocrine pathways. So then also some SNP also affects the PON1 DNA methylation in the promoter. So here you can see the SNP and then the gradual change in DNA methylation of the PON1 gene. So in conclusion, we found that the impact, the epigenetic impact of the pesticides is especially visible in children with the PON1 Q192R genotype. So, and we mainly found like major enrichment of DNA methylation changes in neuroendocrine pathways, which regulate the food intake and the energy balance. And we find many of these links of these pathways with cardiometabolic diseases. So, okay, this was one study, like in Denmark, but then in our hospital, we also have a study where they are studying like another metabolic pathway. More particularly, they are very interested in the NAFLD liver pathology, which is a problem in many obese people And they also had an interest to see whether they already had evidence that the people with NAFLD have in general much more associations with pesticide exposure. So here, this was the start of the study. So they looked for incidence of PCB exposure and then links with fat parameters, glucose parameters, and also the NAFLD parameters. But they did not have an idea what could be a possible explanation. And then we entered also in this study. We were interested if PON1 SNPs could also play a role in this study. So what we did is, so we collected genomic DNA from about 800 people involved in the study. And we also measured PON1 in the serum samples. We had liver biopsies of a few of them. And also we have gene expression patterns of a few of the population. So we determined some of the SNPs that we found which could play a role in the obese phenotypes. Then we measured the activity in an enzymatic assay. So we measured lactonase and aryl esterase activity. Then we looked also to methylation regulation of the PON1 gene. So just recently we also finished the EPIC arrays. And then we also have the gene expression data from some of the samples that we also have the DNA methylation pattern. And then we looked with all this data, we tried to find associations of presence of SNPs linked to methylation changes, which are linked to cardiovascular disease or metabolic disease or MAFLD. So again, like in the previous study with the children, we also find correlations that the PON1 expression is strongly depending on the methylation, and this methylation strongly depends on the SNP which is present in the promoter. So here again, so this establishes or confirms the link of the SNP and the methylation with activity of PON1 towards lactonase and aryl esterase activity. And then we also found that depending on the SNP, we also see differences in the promoter methylation. So altogether, we do not know exactly how these SNPs can affect how they change the DNA methylation. This we do not understand completely, but there is some evidence that around the SNP position, there's an SP1 binding site, and maybe the binding status of SP1 could regulate the accessibility for DNA methylation in the surrounding sequences. But this is something which we need to study in more detail. Then we also found that the PON1 SNP status is also linked to the NAFLD progression status or the clinical severity of the NAFLD progression, so that some people with this polymorphism have a high risk factor for more advanced NAFLD pathology or more severe NAFLD pathology. So also the histology of the NAFLD pathology seems to be more prevalent in people with some of this polymorphism. So, and now we also have done the EPIC array of some of the samples, and we clearly see correlations of methylation with the fibrosis stage and the NAFLD stage. And this is also linked to the, we see a difference depending on the genetic SNP. So again, like in the children's study, we see here a very strong association of SNP, a genetic SNP, with a particular methylation pattern, and then with the NAFLD pathology. Also here another correlation that we make with promoter methylation is also linked to the NAFLD staging and fibrosis staging. So although the angle of this study is a little bit different, we were also to confirm here a critical role of a genetic SNP with changes in methylation of PON1. And now we are also analyzing the genome-wide DNA methylation patterns. So we find interesting correlations with lipid metabolism, with PPAR regulation, with mitochondrial regulation, with the circadian clock. And now, the more interesting thing would also be how pesticides can be involved in this regulation mechanism. So, and part of these results have already been published last year in Clinical Epigenetics. But so this was only correlation of the NAFLD phenotype with the genetic SNP and the methylation regulation of PON1. So now we're working more on the link of the SNP, the DNA methylation of PON1 and the other methylation patterns in this NAFLD pathology related to the pesticide exposure. So in the previous talk, it was already pointed out that the epigenetic clock is also an interesting indicator of health. And we also start to look into that. So it looks like NAFLD pathology is suffering from also age acceleration, epigenetic clock age acceleration. So, and we're also trying to find out how we can reverse or slow down the NAFLD progression by dietary factors or by trying to correct or change the activity of PON1 or changing the methylation of PON1. So this brings me at the end of the talk. A little bit much information from different angles, but if I have one take-home message is that in precision medicine or personalized medicine, neither genetic biomarkers, neither epigenetic biomarkers can give an exclusive, reliable answer why some complex diseases are occurring. So, but that in the future we need to look at an integrated omic approach by combining genetic and epigenetic regulatory levels to find possible targets that we can modulate to slow down the disease or reverse some pathologies. So, or to decelerate or slow down the epigenetic clock aging to avoid adverse epigenetic effects of pesticides. So, and that's the end of my talk. So there's many people involved. So for the obesity studies, we collaborate with the medical genetic department of obesity in Antwerp, and also with the Department of Hepatology for the NAFLD samples. Also we collaborate with the lab in Spain, which was involved in the PON1 studies, the enzymatic studies. So for the clinical evaluation of metabolic parameters on fat and glucose and triglycerides, we have also the lab of Luc van Gaal who is involved. And for the PPAR regulation, we collaborate with the lab of Bart Staals in Lille. And then, we are doing more molecular studies with pyrosequencing and the epic arrays and the pathway analysis. So, and that's the end of my talk. So thank you for listening, and I'm open to answer your questions. Thank you.
So, thank you very much for that, Wim. And I hope everybody in the audience now, for the last sort of two and a bit days, we've talked very much about just DNA methylation and just the epigenetics. And the reason I wanted Wim to come along and give this talk, really is the last lecture of the training school, is really just to remind you that everything is working together. The epigenetics is working with the genetics, and it is doing things. And here we had a very nice example, and this is a really, I think in the future these are going to be classic studies where we've taken the genetics, the epigenetics, the exposure, particularly early life exposure to pollutants, and we're coming up with something. Okay, so over to you. Who's got questions?
Thank you, Wim. Maybe you have told, but I missed, how long exposure affects the DNA methylation?
Well, I think the prenatal exposure, I would say that it was maximum 3 months. So between a few weeks and 3 months. So now, yeah, it's a good question because now we try to mimic it in vitro in cell models. But this seems a little bit tricky because some of the pesticides do not dissolve very well in water. So the solubility is a problem. Then now we're also trying in zebrafish models. But yeah, zebrafish is also swimming in water and the pesticides and many are hydrophobic. So it's really difficult to do this dose response studies like in a cell model or in an in vivo model. But yeah, so for the moment, I do not know the optimal dose or exposure or window which is needed to have a particular epigenetic response. So now it's more pure association studies. And we do not have very accurate measurements also from the pesticide levels in the women which have been exposed. So it's a pity. So we only know by questionnaires that they have been working in the glass houses, but we do not have an analytical numeric value on what is the exact concentration of a pesticide which was circulating for a few weeks or months in this pregnant woman. And then in the other study from NAFLD, so we have they have exact values of the PCB. So I do not know them by heart, but it's rather low concentration. But there we do not have the information on what is the duration of the exposure. So there it was also not in the prenatal setup. It was more the exposome study. So like just an obese population, and they look what are the PCB concentrations in the obese population. And they looked more to the PCBs from another angle, like they looked like people which were losing weight. Then they see increase in the PCB levels in this participants, and this is because many PCBs are stored in the fat. And if you lose weight and you burn fat, some of the PCBs which were stored in the fat are systemically released, and then this high concentration of PCBs can again give some epigenetic effects which can trigger obesity. And so it's like yo-yo effect. So each time you try to lose weight, the PCBs are a little bit going up and give some epigenetic effects which gave instructions for fat deposition, and it's going like, it's like a yo-yo. So the angle was a little bit different there. But since we are living in Belgium, so we have also quite some chemical industry in the cities and also in the harbors. So recently they found huge concentrations of pesticides in the bottom. And yeah, that's why the people are now getting worried. Also for the families which are living in this polluted area, what is the long-term consequence for the children, for the cardiovascular health, the metabolic health, neurodevelopment? So yeah.
Just a quick check. Have you any observation about the obesity is higher in that cities?
Well, I do not have.
This is just observation. You can realize.
So I should ask the people from the epidemiology department if they find clusters of obesity around the polluted area. So this would really be a strong evidence, but I do not have this data. So the information is a little bit fragmented. And yeah, it's also, I think people have been looking at in that, but it's also difficult. So where are the people living and where are the people working? So you can be exposed during work, during the day, or where you're physically living, where do you spend most of the time? And also for the children, where are they going at school during the day? So it's also difficult to know what is the prenatal exposure and what is the exposure during life, because if you grow up in a polluted area, it's difficult to know what has already been accumulated from the pregnancy and what is the new accumulation in the first 1,000 days of life.
Okay, but I think I've got a question for all of you in the room. Can you actually see now? Yesterday we were talking about designing epigenetic studies. And one of the things that was coming through in your small group design exercises was collecting as much metadata, additional data as possible. Can you now see, just where you're living, where you're growing up? It's what you're exposed to. And this is going to be affecting the epigenetics as well. And I think that's one of the things I wanted to bring out with Wim's lecture, is you can't just focus on one narrow thing and ask 3 questions around. You need to try and control for as much as possible in the environment. Because epigenetics, and Wim will agree 100% with me on this, it's a record of the environment in its totality. Okay? Is that clear to you all here now? Okay? So, any more questions for Wim? Because I'm afraid, Wim, you're the last lecture before the coffee break.
Okay.
And people heard the coffee coming into the back of the room not that long ago. So all that is left is for me to say thank you to you, Wim, because it's a very complicated subject, and I think you managed to bring everybody along with you. So thank you very much.